slide is going to just use a liquid sample. For example, we have some gooey pond water, which you want lots of dirt and gunk. So you add 2 drops to the slide. Now the slide is the big glass thing. The second thing, second step in making a slide is using a cover slip. And you want to make sure you don't trap any air bubbles. So you take the cover slip and touch the edge of it to the liquid and then slowly drop it. And that forces all the air out. If you want to, you can use a paper towel to suck the extra liquid out, just so that it gives you sort of a flat slide. So that's your first basic slide. As you can see from these pictures, there's more to hair than meets the normal naked eye. Under the microscope, you can see 3 parts of hair: the cuticle, the cortex, and the central medulla. All right, today we're going to show you how to prepare a regular slide of either a hair or pond water item. So Tak is going to select the slide. Perfect. There we go. And he's going to use the tweezers to put the specimen on the stage, or the slide. Sorry, you get the terminology. So this is your hair, right? Yep. Oh, yeah. And you might want to put the root. Try to get the root right in the middle of. There you go. Oh, nicely done. Now, without a couple drops of water, the hair will come off the slide. So we'll get you to use a dropper. And you can collect a couple drops of water, and you're going to put it on the root of the hair. That looks beautiful. Now you want to add the cover slip. And like we showed you in the video before, you want to touch the edge of the cover slip to the water. There we go, and slowly let it down. And there, oh, beautiful. And in the middle of the cover slip. There we go. That's perfect. Nicely done. Slide we're going to prepare, and we're going to dye with methylene blue. Now this works really well with DNA, so if you were doing animal cells that had DNA in their nuclei, you would use methylene blue. Now when you dye something, you're going to add a drop of methylene blue at the edge of the cover slip, and then we're going to suck it through to the other side. So add it to one side of the cover slip. Probably the side closest to the pond water would work, because, yeah. Yep. So along the edge, anywhere there. Oh, beautiful. Look at that. Is that diffusion or osmosis? So now you're going to take a chunk of paper towel, and you're going to touch it on the other side of the cover slip from the dye. So you're trying to suck all the liquid through so that the dye covers the specimen. So sometimes this is kind of fiddly, and you can figure out how much of the cover slip either. And anything that's in it, anything that's clear will absorb the methylene blue, and you'll be able to see it under the light microscope better. You probably could use gravity if you want to tilt it towards the corner that you're sucking. You can pick up the glass slide and tilt it as well, and that helps the dye go through towards the side that you were, yeah. So. And actually, you probably have enough to work with right there, so we'll go on and look at our slides that we prepared under the microscope.